Hi, my name is Dr. Christopher James Ward from the Kansas University Medical Center. Today I'm going to talk about screening hybridoma clones for monoclonal antibody production. Now, we've just done a fusion between a myeloma cell line and B cells from a vaccinated mouse, and we have several hundred hybridomas, and some of those will be producing antibodies. So we have to screen them for clones that are making antibodies to our particular antigen. And the technique I like to use is high throughput Western blotting. Now, if you want antibodies for fluorescent activated cell sorting or immunofluorescence, it might be better to use those techniques for screening. Now, I'm going to use a manifold to screen 24 monoclonal antibodies per Western membrane. So after hybridoma fusion, we often have 1200 wells. Not every well will have growing hybrids. On average, there are about 600 wells with hybridomas. These clones appear in two peaks, one between days 10 and 11, and the rest from about 12 to 14. The early clones are often IgMs and therefore of low affinity. The IgG1s, IgG2s, and IgG3s, and occasionally IgAs come usually between days 12 to 14. Now, having said that, we've got to screen everything because sometimes you get a high affinity clone early. Now, sometimes I do an experiment like this and we don't get a clone, and that's about a quarter of times, about 25% of time. Usually I get 2 to 3 clones, and sometimes I'm lucky and get 5. So we need a technique that we can use to screen about 200 clones per day. The maximum I can do is about 400. So to do 200 in a day, you need about 8 Western blots in order to do this. And on a really busy day where you've got 400, 16. So it's quite busy when you're screening them. So how do we do this? Well, we start with a very special kind of gel, one that has a single large well going across most of its width. And so in this case, this is a 4% stacker, 6% acrylamide gel that we're using to screen for antibodies against polycystin 2. So what do we do? Well, first we need a source of protein, and what I like to do is I like to get a full-length cDNA for the protein that we're interested in and put an epitope tag on it. And this can be a flag tag or V5 tag. In this case, it's polycystin 2, and we have a GFP tag at the N terminus. So this construct with DNA was transfected into 293 T cells that transfect very easily and make a lot of protein. We then isolated a membrane prep, and this membrane prep is then mixed with what we call blue juice, which is LIDS, lithium dodecyl sulfate, and TSET, heated at 65 degrees and then run on the gel. So we just load it into the big well at the top, and we'll see this once we get into the lab. We run it, and usually you get 1 band of protein at a given molecular weight. Now in our case, we'll have a doublet, which is the dimer for polycystin 2 and the monomer. And sometimes your protein degrades a little bit, and that gives you a characteristic fingerprint when you're going in with your epitope tag antibody. You get a particular banding pattern. And if you see that with one of your hybridomas, that's very good evidence that you've got a monoclonal antibody there that's worth going after. Now, once you've run your gel, you Western blot it. It's just a standard transfer onto nitrocellulose or PVDF, depending on what you've got. And then you put it into a thing called a manifold. We have a mini blotter 24. So it has a plastic backing onto which you put your Western blot. Now you have to block the membrane in milk first, okay, because you're going to apply antibodies to it. And then the top manifold comes down and comes into contact with the face of the Western. And this manifold has grooves cut in it which actually contact the Western blot. And at either end there are ports. So you can inject a protein, you can inject an antibody into the channels, and it will come in contact with the gel from the top to the bottom, including your protein. Okay. So if we put epitope tag antibody in here, or anti-GFP antibody, it will see the protein here and stick to it. Now, we're going to load hybridoma supernates into these other channels, and most of those will be negative. But occasionally you'll get one that does bind to the protein, and you'll be able to detect that. And so what we do is we load in our hybridoma supernates and our controls, and we leave it for half an hour. And then we suck out the channels, we wash them out, and then dismantle the manifold, and then just treat it like a Western blot. Now your secondary is just going to be a global anti-mouse antibody. So we use a goat anti-mouse heavy and light peroxidase that's labeled secondary. And so if an antibody, say, is sticking to the protein here, the secondary will see it, and that will be coupled to horseradish peroxidase, and you'll be able to detect that with luminol and hydrogen peroxide. And so here's an example of one that's finished here. As you can see, here's the control. And in this case, we used a known antibody to polycystin 2, D3, at 1 in 1,000. And here's a positive clone. Okay. Now, as you can see, there are these grooves imprinted onto the Western. You count these across, and this is in position 19. So we can go back to that clone and rescue it. Now, it's important to subclone this positive clone twice. And the reason for that is that the myeloma sometimes survives the initial selection in aminopterin. And if it does, it'll overgrow your clone. And so what we do is we dilute these cells, 1 in 10, 1 in 100, and 1 in 1,000, and put them out into feeders cells again on 96 well plates. And usually in the 1 in 1,000 dilution, you have single wells that are becoming yellow, and you screen those, and you pick the best, of course, and then you screen again. Okay, you dilute it and then subclone it again, and if you do that, you'll rescue your clone, and there should be no myeloma contamination there. Now, I think that this was one of the major problems that people had initially in hybridoma screening, is that they didn't subclone it aggressively enough. And so the myelomas actually overgrew their monoclonals. And so initially you would have a really good antibody, but then it would peter out with time. So what reagents do we need that are kind of specialized? Well, first of all, we need a mini blotter, 28, and we get that from Immunetics. And I've got 4 of them, so that means I can do, because you can do 24 and 24, so I can do roughly about 50 per manifold. And so if I've got 4 of them, I can do 200 at a sitting. We need Novex cassettes, 1 millimeter, empty ones, so we can pour our own gels, and those wide combs. And then we need a goat anti-mouse IgG, heavy and light, horseradish peroxidase, and the catalogue number for that from Southern Biotech is 103705. Now we'll go in the lab and we'll have a go at screening these antibodies. So here we have a 96 well plate that's got some clones in it, and as you can see, we've picked some before. Okay, so this is a plate that has had some clones picked before, and what we're going to do is just pick the yellow ones. Okay, and then when we pick them, I'm going to shout out the coordinates. I'm going to transfer them to a fresh plate. Okay, and I'm going to shout out the coordinates to my friend here, who is then going to say, you know, so supposing we take 1B1, we'll put it into position 1, and then, for example, let's say we go for 6F, so if we go F6, so it'll be 1 F6 in the next one, and all the way along. Okay, so we know the order. Okay, for the manifold. Hi, here's one of the gels I was talking about, and as you can see it's got a single very large comb here. So let's just take this out here, and there's the comb. It's a big one slot comb, and then it leaves behind a big single well which we're going to load the protein into. Okay, and then the protein will be run down the gel, and then we'll stop, and then we'll transfer it onto a membrane. Now the important thing is that you need a one huge slot so that there's a band of protein running across the gel, and then each monoclonal can then come into contact with that band. So these are the gels with the big slots in them. We're just going to wash out the wells very quickly like this, so get a good start. So it's the buffer is tris glycine, and the gel is a tris gel with a stacker, 4% stacker and a 6% running. So I'm just going to load 150 of the protein in. It's a bit tricky, but it's like this. Just bring it along. And do the other side. And that's it, that's what it looks like. Okay, so thank you. And this is how the gel looks when it's running. You just get one band going right across. Okay. So here's the next step. We've got the membranes on the plastic backing. Okay, and then what we're going to do is put our manifold on top like this. It fits like that, and yeah, they're pretty good actually. This one just needs a little bit of modifications, pull back just a little bit. So I just got to screw them in, finger tight of course, don't put them too tight or it can be quite difficult to undo it. Now what we're going to do is put antibodies into each one from our plate here, with a control of course, and the control is going to be antibody D3 to polycystin 2, because this is what we're looking for. We're looking for polycystin 2 monoclonals, so that's 3 and 4. That's what it looks like in the end. Okay, so what we're going to do is we're going to load the manifolds, and as you can see there are these grooves going across the membrane, and first we've really got to put on a control. So the control is 100 microliters of an antibody called D3 to polycystin 2, because we're screening polycystin 2 monoclonals. Now this is to the C terminus of polycystin 2. We're screening monoclonals to the top domain. And so I go in 1, 2, 3 and start and put in the control like this. Okay, and do it for each of them. 1, 2, 3. So we're just doing 3 blocks at the moment. Okay, and 1, 2, 3 like that. Oops. Bubble. If it's a bubble, you just bring it back and then like that. Okay, done. Now we're going to put some actuals on. Okay, so let's look at the plate here. Here's the plate, and so I just go to the very first one and suck, and then go right next to your control and just inject it like that. Okay, we'll just do another one just to show you, and they're nice and red, and so we can actually see where our samples are. This is just a phenol red from the tissue culture. Okay, so just like that. Okay, and so we just carry on and do them all, and in this case we'll do 69 of them, and hopefully we'll get something. Okay, and so thank you. So here we have a manifold. It's got antibodies in them, as you can see, Hybridoma supernatans, and the control. Now we need to get these out of the fluidic lines here without cross-contaminating. So we're going to use this device here, which is connected to a vacuum pump. Okay, and so one end is in TBS and the other end is on a vacuum, and all you do is just place it on top like this and suck. And you can do the next one just like this and suck. Radio. And then what you want to do is dismantle your manifold and put your blocks in here. and wash them 3 times. Okay, so we'll just do that.